Higher biologists, let's do this past paper question together. Now this one is a scientific investigation one, okay, so it's the experimental design question from the paper, and this investigation was carried out into the effect of substrate concentration on the inhibition of catalase by copper nitrate. This is a classic experiment done at Higher, quite often used for assignments. The catalase breaks down hydrogen peroxide into oxygen and water. 10 centimetres cubed of catalase solution was mixed with 10 centimetres cubed of copper nitrate solution. So I'm just going to circle those values there, okay? And a paper disc was placed in this solution for 10 seconds. The disc was then dropped into the hydrogen peroxide. When the disc reached the bottom of the beaker, a timer was started and the time taken for the disc to float to the surface was recorded. Oxygen produced during the reaction caused the paper disc to float. Okay. So I'm going to circle that as well. That's another value it's given us here. This was repeated using different concentrations of hydrogen peroxide, okay? So that's good, repeated. That means improved reliability. A control experiment was carried out. That's good. That allows us to compare, okay? Just to remind you guys, there's reliability repeat, and there is COCO. Control experiments are for comparison. It was carried out where the copper nitrate solution was replaced with water, okay? Again, hopefully from that 5 we know about reliability and controls, but just as a reminder there. We've got the results in the table. We can see the different concentrations of hydrogen peroxide, the substrate, and the different copper nitrate solutions there, and the effect that the different substrate concentrations have on the inhibited catalase, and then we've got the catalase in its own without any inhibitor as well. Okay, so let's get to the questions then. Number 1 says, name the type of metabolic reaction catalyzed by catalase. Okay, as you can probably guess from the nature of the words used in the experiment, catalyzed by catalase, this is a catabolic reaction, okay? And the reason why is because we're breaking down hydrogen peroxide, okay? Breaking down hydrogen peroxide into oxygen and water, okay? The clues in the passage there. So it's a catabolic reaction. Okay, state 2 variables not already mentioned. It should be controlled to make the investigation valid, okay? And we said and COCO earlier. For validity, we remember VAVA. Validity is to do with the variables, okay? So we need to keep those variables constant or controlled. Now, what are some of the variables we can mention here? Now, because this is an enzyme sort of chemistry, chemical-based experiment, the main factors we should be looking out for are MTVCP, okay? If you can remember MTVCP, that will help you remember the different variables there, okay? So that's going to be mass, that's T stands for time and temp, okay, temperature. V stands for volume, C stands for concentration, and P stands for pH, okay? So if we can remember those, that's going to help us out a lot. Now, what have we been given in the experiment? Because it says not already mentioned. We've been given the volume of the hydrogen peroxide, the volume of the copper nitrate, the volume of the catalase. So basically volumes out. It's given us all the volumes. And it says it's placed in the solution for 10 seconds. They've timed it and so on. So time's out as well because they've mentioned that. But what have they not mentioned already? Well, T also stands for the temperature. Okay. Now, at Nat 5, you can get away with just saying temperature on its own. At Higher, we're expected to say the temperature of what, okay? So always say of, the temperature of the solutions, okay? And it would be acceptable to say the temperature of, like, and then naming the solutions, you know, the hydrogen peroxide, the catalase, the copper nitrate, etc. But just solutions covers them all, okay? On that same thing, we could also say the pH of the solutions as well. That would also be an acceptable one. In addition to that, the concentration. Okay, now this time we wouldn't necessarily just say the concentration of the solutions because we have to talk about the concentrations of the individual things, okay? So it'd be the concentration of the catalase, the inhibitor, which is what, copper nitrate, or what else will we put in there? The catalase, the copper nitrate, and the hydrogen peroxide, sorry. Oh no, in fact, we can't say the hydrogen peroxide because that's the thing that we are actually changing, so that wouldn't be one that you could say there, okay? So the concentration as well for that one. There are lots actually you can put for this one, but now that I'm looking at the marking instructions, you can say things like the size, the thickness, the mass of the disc would be fine, or the size, the diameter, the beaker, the same person timing to avoid variations in human error. The same timer or stopwatch as well would be acceptable for this, as well as the same source of catalyst. So anyway, lots of examples there, but by using the ones there, we've got we've got 4 options there by going through this little list here that I've got for you. So do try and remember that one for your exam, okay? And the way I teach my students to remember it is to remember Mr. White's favourite channel when he was a kid growing up in the 80s and 90s was the MTV channel, the music television channel. It was one of the few ways that you could listen to like music on the TV. And I know there's 1,000 channels now, but that was one of the few ones then, and I loved MTV. So anyway, if you remember MTV channel, that'll come up with 4 of those variables, and that's a good starting point for these, especially enzyme-based experimental design questions. Okay, moving on to part 2 here at the bottom. It says, state the purpose of the control in this investigation. Now, the reason why it says in this investigation, because we know that the purpose of a control from earlier on, control is for comparison, okay? If you were asked to give a general definition, it would be to compare the experiment both with and without the treatment, the thing that you're investigating, okay, which in this case is the copper nitrate. So what's the purpose of the control in this experiment? Okay, again, we're going to say to show or compare the results. Okay, we're going to compare the results, yep, both with. Now the thing that we're investigating here is the inhibitor, both with, inhibitor, and without. Okay, so that's the purpose of that. That makes us able to show that it's actually the inhibitor which is having an impact on the results. Okay, this proves that the inhibitor is the thing causing the inhibition of catalase, because when we take that out, we can see that we get a much faster time for the disc to float, and the rate of reaction is obviously faster there. Okay, so that's part 1 of this question. Part 2 here asks us to draw a grid, a line graph on the grid. Okay, so we've got a line graph. That's the first thing to look out for in graph questions, what type of graph you need to draw. And this time it's to show the results using catalase and copper nitrate. Now, we've already got something on the grid here which gives us a bit of a clue, and that's that we've got the results with just the catalase solution, okay? Now, what we need to do is we need to put down the time for the disc to float to the surface, and we're also going to make sure we put in our units so that we get our mark here, which is seconds. Now, don't be tempted to abbreviate that to S or SECS, SECS, just to, for convenience or time. Write out the full thing. Just to avoid any possible doubt about you getting the mark, I would just write out the full thing, okay? Similarly, down the bottom here, we're going to say the concentration of the hydrogen peroxide. And again, just write out the full thing. No point in abbreviating and potentially losing a mark. Okay, I probably should have started that further along, but never mind. And that's obviously going to be a percentage, isn't it? So that's the units there. Okay, now amazingly, you're already halfway to getting your mark here. You obviously need to now work out your scale. Now, if I have a look back at the results from the one they've given us, the catalase, okay, I can see at 0.5 it's at 120, okay, and that's the highest one there for the catalase solution. So I know that's going to be 120. Okay, and that's going to be 0.5 in terms of percentage. Next one at 84. Okay, so if that's 84, and that's 1. 36 was at 2. Okay, so if my scale's going up appropriately, that should be 2 there, and that should be 36. If that's 36, that's going to be 40, isn't it? Next one is at 3, which is 28. So I'm guessing that's going to be 20 on that basis, and that's going to be 0, obviously. That's going to be 0 there. And the next value on our table there is 5, okay, which is going to be there. And that means that must be 4, okay? Now, when you're filling in the graph, you've got to make sure you put in all of the values ideally that are in your table. Okay, just pop them all on the scale and just make sure they're going up at even intervals there, like that one is. So if we